Hey, let's go ahead and get started. We are streaming this as we always do, and I will monitor the Twitter feed to see if anyone out there wants to ask any questions, like I did when I tuned into the conference when I was in Riyadh, Saudi Arabia, a few months ago. So today is the first of our summer intern presentations, where all of the summer interns get to tell us, and I guess really the whole world, since it's being streamed, what they did during their summer vacation. And this will also be recorded, so they can also point other people to show where they did what they did for their summer vacation. As is usual, I'm going to let the mentors introduce their interns. So I don't know who is going first. The Vishnu. Okay.
So hi, I'm going to be really brief. I'm going to introduce to you Uyen, who is my intern for the year. She's one of our brilliant students and interested in a career in informatics.
So,
You can flip it on your.
this. So, hello everyone. Today I will present the project I'm working with Dr. Mohan and our research team during the summer. It is building a better database, how improving data storage can enhance EHR simulation research. So a little bit about myself. I am a senior at Portland State University. I major in computer science with a minor in biology. I'm interested in bioinformatics, machine learning, and human-computer interaction. So this is the outline of what I'm talking about today. First, I will talk about the big picture of our team's project, the project goals, our projects, and data collection. Then I will talk about my role in this project, what I did, my contribution, and what I learned. To begin, let me give you a background about health information technology, or HIT, in general, and electronic health record, EHR, in particular. As we all know, the HIT and EHR are becoming more and more prevalent throughout health organizations in our world today. They have been expecting to increase the overall quality, effectiveness, and safety in healthcare, comparing to the paper in the old system. However, recent researches have shown that EHR is the source for new kinds of medical errors, including the data fragmentation, data redundancy. Those things lead to the failure to identify concerning clinical trends and other medication issues. The problem becomes even more serious in ICU environment, where the amount of data generated every day is extremely high. In average, there are more than 1,300 data points generated each day for an ICU patient. Besides that, there are a large amount of redundant alerts, which leads to alert fatigue and makes the situation even worse. So our goal for this project is to make the EHR safer by first, we assess EHR interface through simulation in the ICU. And second, we establish an educational curriculum to improve EHR use. This means, as Dr. Vishnu usually says, we want to build a better mouse and a better mouse trap at the same time. We solve the problem of EHR safety by improving both the user-friendliness of EHR interface and the training process for EHR use. For our methods, first, we use EHR simulation to replicate clinical scenario and cognitive load. We built up a 5-day ICU case in which we embedded several medical issues, which we call the traps. We then performed the test on 38 participants, including the interns, the residents, and fellows. Each subject is given 10 minutes to review the case in EPIC before they are required to present the patient of the routing. As of the routing. Finally, the subject is scored based on the number of traps they can recognize. Second, we use eye tracking hardware and software to study subject interaction with EHR. Those tools allow us to keep track on where and what the subject is looking at. We employ 2B Studio, which is the software associated with eye tracker. It allows us to capture the gaze event, mouse clicks, key presses, and other kinds of interaction events. Besides that, we manually record the screen change and screen view during interaction. So this is an image of a Windows in 2B Studio. It is recording the subject's eye movement across the screen. You can see its red circle indicates a fixation event. This is the eye tracking recording after 3 minutes. There are more than 300 fixation events happen during this time period. To give a better visualization, 2B provides the heat map, with the color red, yellow, and green indicate different intense levels of the fixations. So, besides providing visualization tools, as showed in previous slides, 2B also allows us to export data into Excel files. The data exported contains the gaze event, mouse click events, key press events, and other kinds of important information. Because to be captured the event once every millisecond, the data exported is extremely large, and therefore it requires a highly proficient managing method in data storage, data retrieval, and data analysis. However, using Excel to store data has unavoidable shortcomings, such as manual entry and update, data relation in a normal form, and the problem of data scattering. These issues prevent us from managing data in a robust manner. So now this is an example of a relation schema in a normal form. As we can see in the red rectangle region, the same information of a person repeated twice, and in some cases it can be more than twice. This makes the database vulnerable to mistakes during modification and update. Data scattering refers to the problem when our data is spliced into many scattering files and stored in different folders. This makes the data hard to keep track and easily lost. So my role in this project is to design a database schema. The requirement for the schema is to be in normalized form, has strong data relation, and has primary keys and foreign keys appropriately set up. From a good schema, we expect to have a robust data to avoid errors in data entry, update, and modification. Last but not least, as the database is built up using MySQL, we can employ the SQL language to help with data retrieval and analysis. This is the solution I come up with after 2 months working with the data. What is shown here is the EHR diagram, which reflects the database schema design. You can see there are 6 entities, or we can understand as 6 tables in this database. Each table has its own primary key, color in yellow, as the unique identifier, and the relation between different tables are captured by the foreign key, color in red. The primary keys and foreign keys have avoid data inconsistency, which I will give an example in the next slide. This is an example of data inconsistency captured by our database system. Based on the primary keys and foreign keys appropriately set up, the system is able to automatically detect inconsistencies, errors. It will raise an alert and require the user to fix this error before they can move on. Now let me talk about the benefit of a normal form database. This is an illustration of a normal form database. The upper table is the patient demographics and the lower is the scoring. In a normal form database like this, only participant ID can repeat more than once in the scoring. All the information of the participant, such as the name, the year, and the program, is stored separately in the demographics table. This allows any entry or update or modification of a participant happen only in the demographic tables and is independent of the scoring. Finally, what I learned in the last 2 months, I tried a research group and gained valuable experience in conducting research. I had a good opportunity to learn about the EHR and ICU through ICU observation. Besides that, I can familiarize myself with eye tracking and 2D studio, which are very useful tools in research. And finally, I had a chance to enhance the skills in Excel and MySQL. Last but not least, I would like to give thanks to my mentor, Dr. Vishnu Mohan, the research team, Dmice, faculty, staff, and my fellows interns. Thank you. So, any questions?
Don't all rush at once.
Yes, please.
You probably have to repeat them too because we don't have the microphone.
So I was just wondering, were there any challenges in designing the database schema?
So the question is, what is the challenge of designing the database
Things you
schema?
didn't expect that, in terms of things you had to abstract out, or relationships that maybe were implicit in the Excel file or in the use case, or was it pretty straightforward?
The thing is that because we collect data and stored in the Excel files, there are the problem of data scattering. We store the data in separate files and it's very hard to keep track. Another problem is that sometimes because of the manual entry, we have the problem of inconsistency where the relation ID of different tables was stored by in different representation, and it's hard for us to keep track and very hard to move on into the next step of data analysis. Does that answer your question?
So I think one thing that I'll add to this is for the learning moment for all of us was actually when we were actually talking to Judy, where our primary assumption was that you would take data and convert it into a better database, and that we could follow simple rules. And Judy asked this question which really got us thinking, and the question was, what are the features that you're looking for? And to me, that was a chicken and egg kind of a thought process, where I always assumed that you would be able to ask whatever questions you wanted to because you had the data in the database, and trying to figure out the feature list or the important features before building the database was almost counterintuitive to start thinking about it because I didn't know what questions I wanted to ask. But as I realized that Judy's way of building the database, of thinking about the features that you require from the database before you even begin to design it, is a much more sophisticated way of, if you build it, they will come, which is what I thought.
So was there a use case or feature that was surprising or that influenced the design of the database in an unexpected way?
Not really. I think more than surprising, it made us kind of focus on the kind of things that we would use the database for. It became a much more functional activity rather than an abstract activity. I think that's important. What we're engaging in is not necessarily, science. It's not just data out there for the sake of data. We actually take the data that we harvest from Toby and convert it and use it to convert the interface into a better way that physicians can interact. So it's actually applied science, it's technology, it's using the scientific concepts towards meaningful patient care. And I think in that context, thinking about the functionality of a database that we would build before we build it makes a lot of sense.
So we should probably move on to our next speaker. Thank you.
So thank you.
So who's the mentor of the next speaker?
Nicole Vasilevsky and Melissa Haendel.
Okay, they're not here.
There it is. Your microphone.
All right.
Go ahead.
Hello, everyone. My name is Ariel Brown, and I am working with Nicole Vasilevsky and Melissa Haendel, who unfortunately couldn't be here today. And I'm working as a part of the Ontology development group. There's a few people here. And this is using mice as models for human disease. So I'm from Maryland. I have the Maryland crab outlining, the Maryland flag outlining the crab because I'm really missing our blue crabs right now. You guys don't have any here, but they're not the same at all. No.
Old Bay's not as good.
Yeah. I attend the University of Richmond. I'm going into my senior year, and I'm studying biology. This is the Gottwald Center for the Sciences. I spend most of my life here on campus, and I have aspirations of becoming a neonatologist. So after my undergraduate, I will go on to med school. So today, I will be talking to you about the Ontology overview, the Monarch Initiative, curation of data, and my results and conclusions. So what is an ontology? So an ontology is a classification system. It can pretty much be about any idea or concept you want it to be. It has a controlled vocabulary. You know the saying, there's like 1,000 ways to say one thing. Well, an ontology makes one way to say that one thing and keeps it unanimous throughout. It's an expressed in a language that can be reasoned across computers. So in ontologies, your terms are given IDs or numbers, and the computers can automatically recognize those IDs between each other. And the relationships between terms are defined. So the way ontologies work it's a broad idea broken down into smaller, more concise ideas. And so that broad idea can be linked to those concise ideas, as well as the smaller ideas linked to each other. And the data within ontologies can be reused. This means when new information is introduced to your ontology, it can form new relationships between old information as well as the new information. So these are classic examples of ontologies you might have seen in your lifetime. And so you have the Linnaeus naming system. You've seen this in your ordinary biology classes, and it just has that same idea of taking the broadest idea of a domain and breaking it down into that smaller idea, that species. And another type of ontology is called SNOMED, and this is used in the medical field. It just contains medical terminology used in clinical health care. And so this is the mammalian phenotype ontology. This is what I worked with all summer, and so this is an example of what I've been looking at. And you see you have your main concept, your classes and categories, and then those classes can be broken down into smaller subclasses. And so an example I would look at would be the cardiovascular system phenotype. That's the main idea. And then the abnormal cardiovascular system morphology, that's the class. And then that class is broken down into these smaller subclasses that you see here. Now, what are ontologies good for? So you have all this stuff, all this research, information from different websites. There's just all this stuff just floating around. Ontologies take that stuff, curate and annotate, just organize that data. That curation organizes it and forms the ontology. And then the ontology can be used to do whatever your project has set out for it to do. And in the case of my project, the Monarch Initiative works with two types of data. Well, I've worked with two types of data, genotypic and phenotypic types of data. And so there are two problems that the Monarch Initiative and the ontology system built within it tries to solve. You don't always know what the mutation is, and different organisms use different vocabularies. As you can see here, the zebrafish, this is all describing the same phenotype, but the zebrafish uses the terminology increased weight, while the mouse would say increased food intake, and the human describes that same phenotype as obesity. What the ontology does is take all of those different languages and put it into one term and making these organisms, these models, interoperable between each other. And that's a big goal that the Monarch Initiative seeks to tackle. And also, you don't know what the mutation is from. And so I work specifically with the mouse models. I work specifically with wild-type mouse models, but this is an example of a transgenic mouse model and how their phenotypes are related to human phenotypes. And the transgenic mouse, if you're not familiar, is just a mouse that has been manipulated. The researcher has either deleted genes or added genes, forming some type of mutation. And it's important to not only use the transgenic mouse within the Monarch Initiative, it's important to compare those results to the wild-type mouse, which I worked with. And the wild-type mouse is basically just the mouse whose genes are unmanipulated, they're born with them, and you use that, and they have phenotypes and diseases as a result of their plain basic genes. And so throughout this project, I had to ask myself the question, while I was looking at the information, I had to ask myself, which phenotypes are a result of background strain variation versus specific directed mutations? And so the background strain variation refers to the wild-type mouse. So there's nothing being manipulated by research in their genes researchers in their genes, and the specific directed mutations are referring to the transgenic mouse. So you ask yourself 3 questions. One is, are the phenotypes or diseases that you're seeing a result of just the background strain genes? Are they a result of the transgenic mouse genes? Are these phenotypes that you're seeing a result of mutations that some type of researcher has introduced? Or 3, is it a synergistic mechanism? Are these genes and are these phenotypes and diseases a result of both the mutated genes and the genes that the mouse was born with? And so I got all of my descriptions from a website called MGI, Mouse Genome Informatics, and I imported those descriptions, which I'll show you what they look like on another slide, imported those descriptions, ID'd them with a mammalian phenotype ontology term, and imported them into the Monarch Initiative website. Now, I was a curator. You might not know what that word means. So to curate is to pull together, sift through, and select for presentation. So basically what I did was pull up all of these descriptions of the mouse strains and just pull out what was the phenotypes, just the important information, all that messy data, and make it into something organized. It's just like a few little posters. It was sometimes really hard to keep calm and curate because there was a lot of information to look through. And so here's an example of the raw data that I looked at. And on the top, the strain is called Bob or BOB. And it has 4 phenotypes that I could pull from and map to MP terms, or mammalian phenotype ontology terms. And so you have albino, small spinal cord, high lymphocyte, PHA response, and develops leukemia on infection by friend virus. And so the albino happens to be mapped to 2 MP terms, and that's absent eye pigmentation and absent coat pigmentation. And all of my annotations were done in a program called Phenote, and Phenote contains the entire mammalian phenotype ontology within it. So you can take your description and just annotate and find whatever term that you needed to find to map it to. And then when you save it, it's saved on a program called Smart SVN or Subversion, and the files are saved to a user interface under version control. So summary of my data. I curated 364 wild-type mice strains, and that's 1,550 annotations. So it was hard to keep calm and curate. So the results. So with the work I did was important. You know, it seemed tedious at the time, but it was important. As you can see under A on the y-axis, these are the background strain genes that are actually involved in a number of these phenotypes that you see on the x-axis. And if we look at part B, you can actually see the number exactly. So for the first strain, C3HEB, it has 17 phenotypes that are just being contributed by the background strain genes. And it has 58 phenotypes that are working synergistically, meaning the background strain genes as well as the mutated genes are contributing to that phenotype. So in conclusion, ontologies are a useful way to describe phenotypic data. And by annotating this data, it's allowing us to know which genes or which phenotypes are actually being contributed by what type of genetic background. And we actually found that 1,207 strains were being contributed by the background strain genes in the wild-type mouse out of 60,203. So that's, I mean, it might not seem like a lot, but it's significant when you're doing like research papers. You have to contribute some of your findings to, you know, this research. So, and I didn't just do this one project. I also worked on 3 other projects. I worked on the Journal Decay project with a colleague, Robin Chempo, and we discovered if journals were available in print or electronically, and this is just looking at what happens to journals over time, and we did approximately 403 journals. Another project was the elements of morphology, and we basically annotated images. Instead of with the mammalian phenotype ontology, we used the human phenotype ontology, and that was 148 images. And the Omnia curation is much like the mouse curation that I did, except it just used a number of animals such as cats, dogs, horses, sheep, just a lot of animals. And I did 154 diseases with that. And I have to acknowledge my ODG group, of course, my mentor, Nicole Vasilevsky and Melissa Haendel, and the participating institution, and a special thanks to Dmice and the OHSU library. And this is a screenshot of the Monarch Initiative, so if you want to go on there, my work will be up there soon. You can just kind of click around and find all the links between diseases, phenotypes, genes, and models, and it's kind of cool. So, thank you.
So I presume these annotations are possible connections between the gene and the phenotype.
Yeah.
What's going to happen to those annotations? How are scientists or other people going to make use of them in their research?
It forms the new links between discovering. It's also, this whole project is connected to the Undiagnosed Disease Program, so it's going to form those new links that can help you discover these diseases within humans that are not very well linked to any genes or phenotypes or even models that can help you discover those connections. So, yeah.
I guess we know those phenotypes have incomplete, I'm not sure the genetics terms, but incomplete penetrance where, you know, just because someone has a genotype doesn't mean they're going to get the phenotype.
Exactly.
So I assume there need to be experiments to determine the amount of penetrance or...
Yeah, and that's why what I did was really important, because a lot of researchers might think, hey, I deleted this gene and then I see this phenotype or disease in my organism, but you have to realize sometimes it's not just your action of deleting that gene, it's, you know, the organism could have already been born with that mutation, and you have to make sure that you're accounting for all the information, so, yeah.
Ariel, I have a question about sort of medical literature.
Mm-hmm.
It's, you know, there's an ontology in medical literature called MESH, and I guess you've seen this. It sort of characterizes all these scientific papers.
Mm-hmm.
And, you know, we're supposed to learn MESH and use it to march up and down the hierarchy to find what we want. What I feel like I'm seeing is that people are searching for literature more and more using Google rather than MESH. And so I guess what I'm wondering is, with what you've done, there's a ton of work here.
Yeah.
As things like computing power and search engines get better and better, do you see ontologies becoming more important or less important?
I think they're going to become more important and more like a valuable resource to use, because it's taking that data that's thrown out in all these different places and is bringing it to one central location. So I would definitely use it. I didn't know about it before the summer, but it's, yeah, I would definitely use these tools. Yeah.
Can I chime in on that real quickly? Things like ontologies and controlled vocabularies, finding ways to integrate that with traditional IR is actually a really active area of research in the IR and NLP world. It's, you know, Google most definitely does use that sort of knowledge in figuring out results. So having good curated data can be a really powerful signal for more standard text retrieval.
Great, thank you. So who's our next speaker?
How do we do it?
Yeah.
Okay, there's one, there's two. Come on. Oh boy. You want to do it again? You got it? Okay. This one. I'll come around. Go where? Oh, yeah. Is she sitting here? Okay, I'll just take this. Seems like what we're doing.
Let me go ahead and introduce our next speaker, Katie Abrahams. Katie was here last summer as an intern, so this is her second year back. She's a computer science student at Portland State. And she did a great job last summer, I heard, and she's done a great job this summer. She's working on eye disease and computer-based image analysis for that. I'll stop over here and just let Katie do the rest of the talking.
Sure. Yeah, going into my junior year at Portland State doing computer science, and the summer has been so super cool. I'm really excited to tell you guys about retinopathy of prematurity. Make sure you know how it's set up.
Okay, that's coming on. I think this is these are those. Okay. Computer, no signal.
I think it might just because the computer's still turning on.
Oh, is that it?
Yeah, I think
Okay.
so. Be turning on. We'll get there.
Okay, I got
Still
to log
looking
in.
at
So I have to log it out. It's not in. This thing.
Adapter problem, the wireless. I don't care about the
Oh,
wireless
yeah.
adapter.
You don't need the wireless. Okay.
We saw that before, so that's kind of a good sign. I think you might have to do a connection over here. I'm not
Oh,
sure.
is that how it works?
I'm not sure. You know, like this guy probably knows what he's doing.
Oh, you got it before.
There is a way.
No, I haven't.
Should we try? Oh.
There
Alright,
it
awesome.
is.
Fantastic. All right. Hey guys. This summer I worked with Dr. Chiang to develop a computer-based image analysis system for diagnosis of retinopathy of prematurity. And ROP is a retinal disease that can affect premature infants, and if it gets bad enough, it can cause vision loss or blindness. It's pretty... Let's see. And we hope to eventually develop this whole computer system to provide this quantitative analysis for retinal images to aid in diagnosis. Today I'm going to talk a little bit about eye anatomy, give some reference points for what I'll be talking about. I'll talk about what retinopathy of prematurity is and my role in looking at work with retinal images and data analysis. I'll talk about diagnosis and treatment, and I'll go over a preliminary study that was done to sort of test this quantitative analysis for images, and I'll talk about hopeful project outcomes and goals. First off, a couple images of the eye. I wanted to point out the optic disc on the bottom of the left and the center of the right. It might be a little hard to see on here, but that is where the eye connects to the brain. And I wanted to also point out the retina, where we sort of gather light and what it allows us to see, and that's where the veins are that are involved with retinopathy of prematurity. And I also want to point out the macula over on the bottom left and in the right image on the center right, which is where the highest area of visual acuity is. Introduction to ROP. ROP is a retinal disease in premature infants that can cause, if it gets bad enough, poor vision or blindness due to retinal detachment. And risk factors for ROP, aside from being premature, are birth weight less than 1,500 grams when the average is about double that. And babies born at or before 30 weeks when full term is 40 weeks, and high oxygen levels. And these two images, one is normal, one is retinopathy of prematurity. On the normal, I'm going to point out that you have some, there's no mouse unfortunately, I'll just point to it. You can see that these vessels are very smooth and thin. This is a normal eye, and over with retinopathy, there's thicker vessels and they're tortuous, they're very squiggly. So that is where it's starting to get to be pretty serious. And that eye has something called plus disease, which I'll talk more about later, but it is a pretty severe form of retinopathy of prematurity. And this is a more disease progression or potential disease progression. These are 3 different patients, but it sort of is a good classification. I think up at the top is normal. They are smooth and thin. There's no retinopathy of prematurity in this eye. Over here we've got pre-plus, which is sort of an intermediate between normal and plus. You can see the vessels are getting a little bit thicker, a little bit more tortuous, but not nearly as bad as the plus disease, where you have much thicker. And the thicker vessels are the veins, and the tortuous ones are the arteries. So that is improper development. It can lead to some bad vision problems. diagnosis and treatment. ROP is diagnosed by eye exam by an ophthalmologist, and you can see it up there. And images like the ones I worked with, and like I'll show you in the next couple slides, are sometimes taken, but not always. They're not used in every single diagnosis these days. And treatment and a little bit of history. Prior to the 1950s, retinopathy prematurity was not known about because premature babies didn't survive. Now, from the 1950s to about the 1980s, we knew about ROP, but there was no treatment for it. And in the 1980s, we undertook, there's an extensive study that was undertaken, and a lot of work for it was done at OHSU, which is pretty cool. Joe Robertson did some work on it when he was starting his career, president of OHSU. And they actually found a treatment that worked, this cryotherapy, this freeze therapy that was developed. And they don't use that so much these days. They've sort of developed this laser therapy, but they both work on similar principles, where you will, and I showed some laser treatment over on the bottom here, they will kill the vessels in the periphery of the vision in order to try and preserve that central vision near the macula and the optic disc, and hopefully stop the progression and allow the eye to develop normally and preserve as much vision as possible. And you can see the laser scarring on the right, but the vessels over here, they've regressed back to what looks much more like normal. They would have been much more tortuous and thick and squiggly and bad before. They also have an under development and is being used in clinics. They have an injection, a drug they can use to hopefully stop the progression or at least slow it more realistically and allow the eye to develop normally and not quite so badly. And there is one important point. In the diagnosis of retinopathy prematurity, it's not clear-cut. There's some diagnostic variability among experts, and that can cause some problems because this diagnostic variability determines how you monitor and treat the disease. I'm going to talk a bit more about that here. So this was something that was done, Dr. Chiang told me about this study, where they gave these two images to experts, really world experts, to see whether they could diagnose as plus or not plus. And plus disease is the most important parameter when it comes to diagnosis because more than any other thing, it will, or any other aspect of the disease, it will determine whether or not you require treatment for ROP. And so they really want this to be well diagnosed. And in this one, you can see that this one, 14% said it was plus, 86% said not plus. But on the image on the right, there's even more variability. Or 77% said plus, but 23% said not plus. So it would really determine what you did as far as the treatment for that. And this sort of variability, this and some other things, were a large part of the motivation for designing a quantitative analysis system, which is what I worked on and what the whole project that I worked on is about. And that relates to these images. So these images, and this one and the one on the next slide, show what I worked on for a lot of the summer, where I took an image up on the upper left is a RetCam image, and the RetCam is the tool that they use to take these retinal images. And then I, in Photoshop, use separate layers to manually segment, trace these vessels. First I trace the veins, separate layer for tracing the arteries, and then I combine them all into one layer and also trace that optic disc, which connects it to the brain, in that and sent it off for diagnosis. And I'll show you another one as well, but this one is for plus disease. Same method for segmentation, but this one, and you can see in the plus disease, vessels are much more thick and tortuous. This one, it would be treatment requiring. And data analysis, the next step we're working on currently. And so the images, like the last two slides, we sent those off to an awesome team at Northeastern to deal with them. And they're starting to, we just got the results back this week from the initial findings. And we wanted to compare my segmentation, the human, which we treat as the correct segmentation, with a computer segment image. And they use an algorithm in MATLAB to do that. And we want to test what portion of the image is correctly segmented by this algorithm. And I'll show you on the next slide some comparisons between the two. And to do that quantitative image analysis, we used a couple metrics for that: sensitivity, specificity, and accuracy. And sensitivity measures the true positive rate, which basically means, did it segment when it was supposed to? Is that white tracing there? So false negatives in their algorithm decrease the sensitivity, and that happens when the algorithm doesn't segment when it should. Specificity measures the true negative rate, so it correctly does not segment, the image is left black. And they only tested the part of the image that showed the rectum. There's some black in the corners, but they ignored those because that isn't an image of the eye. And for the specificity, the false positives decrease the specificity, and that happens when the algorithm segments something when it shouldn't. And they also use accuracy, which is the portion of the total image that is correctly identified. I'll show you some images here. So the top left is the one image that I segmented when all those layers are combined together. So that's the final segmentation. And on the right is the algorithm segmented image. They used that same RetCam image and fed it through, and they compared it to my segmentation. And you can see that it's pretty close. It's not perfect, but it looks pretty good on that top left. Bottom right is a tree structure that was also automatically generated by the algorithm. And that measures the branching and the junction points in the images. So it automatically generates this tree to either do, you can see the different colors based on where the junction points hopefully are. Or between two junction points, or between a junction point and the edge of a vessel, the end of a vessel, I should say. This table measures the segmentation metrics and percentages for the averages, because we gave them, I think it was about 100 images total, and all those images had this gold standard diagnosis, gold standard agreement between 3 experts. And so we had a bank of normal images, a bank of pre-plus images, and we also had plus images, about, I think it was mostly normal, maybe 30 or 40, and then 15 about of the pre-plus and plus images. And the sensitivity, which was when it was segmented, when it should, was white vessel tracings. That's about 60-50%. Specificity and accuracy are much higher. Specificity is when it correctly does not segment. So that's 90%, high 90s for all those. And the accuracy, that overall segmentation metric, is also very high. And that's to be expected when the image is mostly black, to have the specificity and accuracy be higher. And according to the team at Northeastern, it's okay. They'd like to make some the algorithm improvements. But as far as initial findings, I think they're pretty happy.
What was your underlying disease rate?
Sorry, what?
When you look at your population disease rate prevalence.
Sorry?
The prevalence of disease in these populations.
Oh, yeah, as far as the amount of babies that get ROP?
For the sample. What percentage of them have retinopathy or that?
Oh, sure. The normal, so the question was, what percentage of these babies have ROP in these images? Thank you. The normal that wouldn't have it, pre-plus would have had ROP, and plus would have had severe retinopathy prematurity. So all these would have had retinopathy, all these would have had retinopathy, and this would have been a normal comparison as far as image segmentation. Does that answer your question? Uh-huh.
So how many images, I think maybe what you were doing was how many images were there, and how many were normal, how many were pre-plus, and how many were plus? Within the sample.
Sure.
Was it like 30, 30, 30, or was it 5 normals and then 10 average and 5 plus, or what was the?
Oh yeah. I think of the roughly 100, I think it was about 75 or so. I don't remember the exact numbers, but the majority were normal. I think it was probably about 35 or so, and then we had less for the pre-plus and plus, and I don't remember the exact numbers, but it was probably about 15 of each, something like that, so smaller numbers. And I should say, as far as population in general, I should have mentioned this earlier, but about 50, about 50% of at-risk babies, the babies that were born, the metrics I gave for low birth weight, high oxygen use, that sort of thing, but 50% will develop retinopathy. This is in the general population, not in the images that I did, but they'll develop some form of ROP. Of that 50%, about 90%, it'll be mild or moderate, which I believe pre-plus would fall under, though perhaps not. He knows we're talking about him. About 90%, it'll be mild or moderate, and it'll disappear on its own. They monitor it in clinic every 1 week, every 2 weeks. But the 10% will develop this severe treatment-requiring ROP, laser or injection, usually laser. As far as the general population, about 2/3 of premature babies will get some form of retinopathy prematurity, and in the general premature, I think in 2013 it was 1 in 9 babies were premature. So it's a lot of kids overall that are at risk for this, or that will get it. Cool. Hopefully, does that clear up everything about this slide? All right, cool. So it's a lot of info. Yeah. And this was the preliminary study that I mentioned earlier. This was done with a different set of images, nothing that I worked on. We collaborated with people in New York because we wanted to test to see whether you could do quantitative image analysis and see if it would work and work as far as diagnosis. And so this looked at the retinal field of view, and they wanted to get a standardized field of view because you have these retinal images and they have different views, different ways that they point, sort of thing. So there's not always a standard image. They wanted to get a standard sort of field of view related to the position of the optic disc of the image. So you would always have sort of a standard way of looking at it. And it was a relatively small number of images evaluated for this crop shape and size. And they looked at rectangular versus circular crops. And they also looked at different crop size, so how far you were, what percentage of the image you were looking at based on the total. And they did find, and this is pretty cool, they found that the rectangular crops, which is the sort of area you see there, compared to the circular. And they correlated well with the quantitative parameters for tortuosity and dilation. And tortuosity and dilation were really important because that relates to how severe the plus disease is, and therefore probably whether there's treatment requiring RP. And that was super cool to see, and the study that I worked on is working to validate those findings in a different set of images. And they want to validate that quantitative image analysis does work and that you can get quantitative metrics and it'll work for disease diagnosis, which I thought was definitely super cool. As far as what's next, getting back to the project I worked on, what we're working on right now is comparing this human segmented images to computer segmented to test the accuracy of the algorithm and if improve it as needed to make sure that we can get an accurate algorithm. We're also getting this quantitative data from the vascular and arterial parameters, which is, again, the diameter and curvature of the vessels, important for plus disease. We're working to get feature extraction from the images, and feature extraction is the diameter and curvature of the vessels. And we also want, eventually in the future, to be able to evaluate plus disease based on the algorithms that we're developing, and based on, in the image, the diameter and curvature. And we eventually want to use computer segment images as a diagnosis aid, be able to get a quantitative parameter back from these images and use them as a decision aid. And these three images are examples of quantitative measures, a curvature, length, and angle of the vessels. This summer I learned a lot about retinopathy prematurity. I knew nothing about it going in, and I learned a lot about it. I got to visit the NICU to see the premature babies. That was very cool. I used Photoshop to work with retinal images and identified those optic disc centers, the XY coordinates, which are important for positioning the image and getting that sort of standardized field of view. I learned about the complications of diagnosis. There's a lot of that. I explored data analysis, and I learned about study design for computer-based decision support. Huge thank you to my mentor, Dr. Chiang, Susan, the study coordinator, and the whole ROP team. Also my fellow interns, thanks guys, and OHSU.
I think we want the last speaker to be able to present.
Yeah.
We're running a little late, so maybe people could ask questions while the next speaker is setting up.
Yeah, I'll break this down. I'll leave the mic on for now so I can repeat the questions. Any questions from anybody? Yeah.
I have a question.
Yeah.
So what you were doing was creating a gold standard against which to measure an automated technique for detection of the vessels, right?
Yes,
and
yes.
the vessels can be detected. Is there a measure of tortuosity that's been developed? I mean, if you can detect the vessels automatically, what's important is detecting the tortuosity of the vessels. Is there such a measure?
Yeah, so the question, is there a measurement for detecting that tortuosity? And that's, they do have that, and they can sort of measure right now the angle of the vessel, see how tortuous, they have that tree structure, which is the colored image, where they can measure the branching. And so yes, they have some mathematical ways of doing that right now, and they do have that. And I think the feature extraction I mentioned, they're working to really sort of hone that and fine-tune it. Is that cool? Mm-hmm.
Because, I mean, that has to be.
Yes, yeah, and they're working on that. I know that the team at Northeastern is working on that right now, and I believe and they've done some studies previously where they have measured that, and they want to integrate that into this system.
Great, thank you. So, who's the next mentor? Okay.
So I'm really happy to introduce Haendel Bachrach. Hannah is leaving on Tuesday morning to go back for her senior year at Skidmore College in New York, about 3-4 hours drive north of New York City, where she is an environmental sciences major and a pre-med. So she also had the great pleasure of taking the MCATs this summer. And she did a great project. We had a lot of fun with this. It's something I wanted to do for a long time, and we kind of had to play it by ear, day by day, how to go through this. But he came out with some very good results. Okay.
Can I make it so I can show my notes on this thing? That's all right. I can go without notes.
No notes. Come on.
All right. Hi. I'm Haendel Bachrach. I was working with Judy Logan, and so I will talk to you about computer modeling of endoscopic workflow. So I'm going to talk to you about sort of the background of how we got started on this project, how we collected data for it, how we then modeled that data, and then what the model eventually showed us. Okay, so background on getting started. As I said, I worked with Judy Logan, and she works with a group called CORI, which is a group that has developed an interface for endoscopists to use to input their data from their procedures, and it'll then go to a national database so that if there's any research that is being done on those procedures, all of the data is in one place and easily accessible. So obviously that opened the door into endoscopies, and we worked down at the CHH, so the obvious model to use was the 6th floor there, which has an outpatient endoscopy suite, mostly colonoscopies and EGDs. So that's where that came from. And so CORI collects a lot of the numeric data. It has, the number of colonoscopies, how long they take, the number of EGDs, that sort of thing. So we wanted to look at it from a different perspective. And so then we can get to the goals of the project. What we wanted to do is we wanted to look at the workflow through the suite. So how the doctors actually work through their day, what they do, when they do it, how long it takes. So what we wanted to do is use the suite down in the CHH and observe them to get what their workflow is, be able to model that, and then be able to make it more efficient by adjusting the model. And then hopefully we would be able to apply that to any given endoscopy suite and not just the one down here at OHSU. So then the question is, what is workflow? So workflow can be studied in a number of different fields, but there's not one simple answer because depending on the field and depending on the specific question you're asking, it can be a lot of different things. One paper we looked at came up with this model, or with this figure, that had all of these different categories for workflow. So for example, for what we were looking at, we were mostly looking at temporality and time studies, maybe some design components. But the point is, there's really a lot of different components of what is workflow and what goes into workflow. This study actually came up with one definition that would be in my notes that aren't appearing on the computer. But so pretty much what they came up with and what I sort of think of it as is the step-by-step steps through a process that you can measure in a sort of defined metric. Because you can't just say, oh, well, they go from here to there and they spend some time. You have to actually be able to say, this is the information we have and this is the metric we're using to say where they go when. So once we sort of had that figured out and figured out what workflow was for us, we could start collecting data. So first we had to start with some preliminary observations. And these actually started about a year ago with a master's student, Bachrach. And so what he did is he observed different endoscopists as they went through their half-day long shifts. And so what he started with was making a list of all of the different things they did in the day. And the idea was that if we could record what they did each day and how long, and the order in which they did them, we could make a workflow model. Unfortunately, the list Bayer originally came up with had, I think, more than 60 different activities. And that's just not really feasible to record. You would have to have, a giant data sheet, and it just didn't make any sense. So with even more data collection observations, we were able to narrow it down to this list of activities, which are computer EHR, so that's any time the doctor is working on a patient's EHR, electronic health record. Other computer, so if they're emailing someone or researching someone, so working on the computer but not specifically on the EHR. Other medical, so if they're talking about maybe a patient that wasn't there that they saw in clinic a different day or consulting with another doctor. Patient interaction, so talking to the patient, whether that's consent or if that's just, oh, hi, I'm Dr. whoever, how are you doing? Personal, so eating, going to the restroom, procedure, and so we got the procedure time data from Corey, so they define it as scope in to scope out for endoscopies. Walking, again, pretty self-explanatory. And then sedation, which was actually one of the hardest things to record because we started the sedation count when the doctor instructed the nurses or techs to start sedation, and sometimes I was more of a nonverbal cue, so that was just another area that we felt we needed to have, but was hard to record. But so ultimately, these are the activities that recur for every single patient. They have to happen. So, that is what the workflow is. Personal could be different for every different doctor, or other medical could look different for every doctor, but those all happen. So once we had that, we had to figure out how to actually record that. And we decided to use Excel forms, which are coded with VBA coding, this is what this is an example of. So first I started out struggling through learning that, because the biggest challenge was really that the activities of the doctor can change every few seconds. So we have to be able to get a timestamp that records down to the second, and you can just do in one click. And as you can see, this is the final form we ended up using, and there is a timestamp with one click gives you the time down to the second. So that was something that we thought was absolutely vital for whatever data sheet we'd be using. And then after I figured that out, we had to figure out what this form should actually look like. This is what it ended up looking like, but first it had radio buttons, it had drop-down lists, we tried maybe typing it in. We just had to have something that was quick and would be uniform, because if you said one time talking to patient and another time talking with patient, when you try to analyze that data, it'll come out weird. So in addition to actually programming this and getting it to look right, throughout the data collection, we even changed what data we were collecting. For example, at first, we weren't collecting sedation times, but then we realized that really is an integral part of the process. There is a point where the patient is being sedated and the doctor is either in the room or working on something else. We also, for example, didn't record at first which patient's EHR the doctor was working on, which we also realized was pretty important because otherwise you don't actually know when the doctor has finished working with that patient. So once we kind of got most of the bugs figured out with this, we could move on to the data modeling. With modeling, we had a theoretical approach and what we thought it would look like, and that was very, very different to what the actual model using Arena, which is the program we used to model, looked like. So for the theoretical approach, we wanted to follow the doctor. We didn't want to know what the patients were doing. We wanted to know what the doctor did throughout the day, what their steps were. So we thought it would look something like this. As you can see, the procedure is on the line. The procedure has to happen. There's no patient if there's no procedure. So the idea was that they can take any of the different routes through this as long as they do some things before the procedure, then do the procedure, and then do some other things. They can go in any order as long as walking happens at some point because they have to get around. EHR has to happen at some point, otherwise they wouldn't be recording their patients. So we were thinking it would look something like that. Our very first model with Arena looked something like this. So similarly linear, but there's not all the loops, and the biggest difference was that with Arena, you can't really follow the doctor, we realized. You have to follow the patient. So how Arena works is that the patient arrives, and they move through each of these processes. And each process has a time associated with it, and they can also grab a resource. So for us, that resource was either the procedure room or a doctor. So how instead of following the doctor, we follow the patient, but the patient uses the doctor. So, like I said, this is where we started, and this is where we ended. So they look very different. Because as we went along, we realized that this was actually just more and more complicated. So first off, the 3 branches at the beginning indicate that there are actually 3 different types of patients moving through the suite down at the CHH. There are colonoscopy patients, EGD patients, and patients that are getting a double procedure, so both sort of in one go. So it's important to differentiate those because the average length of the procedure is really different for each of those. Then, you can see the people moving through because Arena has little animated people. But so the next branch is where the room gets cleaned, because down at the CHH, what they normally do is one doctor uses the same procedure room throughout the day. So you can't have one patient go in and then leave and then immediately have a new patient go in because you have to clean the room. So here we have, after they go through the procedure, the patient keeps moving through the process, but the room stays up there and gets cleaned for a while before the next patient can come in. And then finally, down here we have another branch like that indicates that after a patient's discharged, they might be able to leave right away, but the doctor might not still be done. They might need to either do other medical things associated with that patient, so finish up notes, or they might need to do personal things or talk on the phone or whatever it is. But point being, the patient can be discharged, but the doctor is still busy. So So yeah, this is what the final model ended up looking like. And then with each process, like I was saying, there is a time associated with it. And so that's where our observations and data collection numerically came in. So we used the time that, for example, doctors took reviewing patients' EHRs, and we associated that with each process. And also with Arena, we were able to make this model and then have it be run through 1,000 times or however many times you want. So we were sure to get really significant data that, it wasn't just a fluke that this one time this patient took a long time. It was the average of multiple runs, so we could get a really good feel of how long it's taking patients to move through here. So results from the model. So what we started looking at wasn't really, so like I said, we wanted to look at what the doctor was doing. And what I was looking at sort of changed as what became practical sort of made itself apparent. So the first thing I really looked at was just changing the order of the schedule. So D stands for double procedure, C for colonoscopy, and E for EGD. So these are the different schedules. They do about 6 patients in a half day. So, and each different procedure is scheduled differently. For example, double procedures are supposed to take about an hour, colonoscopies are scheduled for 40 minutes, and EGDs for 20 minutes. So then there's a whole conflict with how to make these schedules work in the model, but this is what we ended up with. And in addition to just making the model, which is a great start, it suggested that schedules where double procedures are last tend to be longer for the amount of time it takes a patient to move through the system. And it takes longer for a patient to move through the system when the colonoscopies are not consecutive, so when they're split up, like in this one. So those are some areas that, whether or not the model is necessarily perfect, these are things that it's suggesting that it would be interesting to look at and see if that does hold true. Now, the other thing that we got from the model was that as the instantaneous utilization of the doctor increased, so that means as the amount of time the doctor is being used by patients increased, the amount of time it took for a patient to go through the process increased. So it sounds confusing at first, and I had to look at this graph for a while, but that's actually pretty logical. If a doctor is busier, it takes longer for the patient to get through the clinic. So we thought that was a good indication that this model does work. That's how logic works, that makes sense, and that is exactly what our model showed. So we took that as a good sign. So while we did get the model and a slight understanding of Arena and some of these data suggestions from this, there also was one big shortcoming of the model, and that's that the average time it took for the patient to move through the system in the model was about 79 minutes, whereas when we collected the data, the observed time was about 53 minutes. So that's a pretty big difference, and when I tested the data, it is statistically significantly different. So that presents somewhat of a problem. And it definitely means that the model needs to be worked on. It doesn't mean that you can't use the suggestions that the model is showing. You definitely want to test them before just saying they're right, but it could just definitely be a good place to work from. And also, luckily, there's a lot of areas for improvement in the model which could try to rectify that difference in time. For example, the ratio of the types of procedures is off in the model compared to what it is in real life. There's a higher ratio of double procedures in the model than in actuality. They do very few. It's about a 10 to 20 to 1 ratio, with the double procedures being 1 in real life, but not in the model. And the time distributions were another area for improvement that we really struggled with. So we took a lot of data to try to get those distributions, but using Arena, when they use, for example, a normal distribution, you get the long tails on either end, which aren't very realistic time samples. So we tried to use triangular distributions, which didn't always fit the data very well. The scheduling times, like I was saying, we had a lot of trouble trying to figure out how to schedule things differently on Arena, and I actually have a new form of the model now that has a different method. But so, point being, there's a lot of changes that can happen. 0 times, by that I mean, while we were really careful to make a list of things that had to happen, sometimes a patient, for example, isn't sedated. And if that patient isn't sedated, that sedation time should obviously be 0, which will bring down the whole average. But it was really hard, based on how I was collecting data, to tell if there was no sedation or if I just didn't catch the onset of sedation. Process variability. The doctors, like I said, we tried to make the model variable enough to get all of the different doctor schedules, but we know of at least one doctor down at the CHH that reviews all of the EHRs in the morning before any patients arrive, and that sort of schedule can't be accounted for in our model. Also, the compensation of the doctors, for example, if they see they're behind schedule, they can speed things up, and our model can't do that. So that's definitely something to look at to see, does that ability for humans to, speed things up or slow things down really affect how the model would go. Finally, how we figured out the time for double procedures is by so Corey has the data for EGDs and colonoscopies, but not specifically double procedures, so we just took those two distributions and added them together. But maybe that's not exactly how it works. Maybe when there's a double procedure, they only do that because both of them are short, or maybe they somehow affect each other, so it's possible that that distribution is just off. And then finally, with other medical, I explained how we would have put it into our model, but we actually didn't even have a chance to look at that distribution and put the numbers in. So right now, in the model, this branch is set to 0 time. So there's not even the time added for, going to the bathroom, talking on the phone. So in theory, that'll increase the amount of time it takes a patient to get through the model even more, which would make the 79 to 53 minutes even, more off from one another. So, ultimately, the model isn't perfect, and it's so frustrating because when you look at it, when you walk through the system, it seems like it should be, but the numbers just don't quite work out. But there's definitely a lot of ways to improve it, and maybe Arena isn't the perfect software to be using to model it. It's just, there's a lot of questions to ask. But this is an area that's sort of newly being looked into, so it's a really cool place to be doing research, and it can have big implications if we can actually get the model to work. So that's what I found. Thank you to Judy and Dmice and everyone. But,
Questions?
Yeah.
Did you ever use the input analyzer in Arena to look at different distributions?
Yeah, that is what we used. So Arena has an input analyzer where you can put your data into the system and it'll make a distribution for you. So that is initially what I did is I put it in and you can have it fit the best distribution. So that's what I did. And our numbers are really high like they were in the final one. But then after talking to people who are much more experienced with Arena than me, we thought that sometimes with things like normal distributions, like I was saying, the tails end up skewing the data. So we thought triangular distributions might be the safest just because that's where, you have a minimum and a maximum and like a point in the middle. So we tried to, get rid of outliers so they would be more closely fit to triangular data. But with triangular or with the distributions that fit it the best, the data was still, for some reason, the times are just too long. Yeah. Or, I don't know. Sure, in the back.
I just have a question. Did Arena make suggestions about bottlenecks, and were those consistent with what the staff were observing?
So Arena is really hard to use. The first, 10% of it, as Judy talked about a lot, is really easy to figure out. I got the first model and showed it to her, and it was like, this is easy. And then, we turned it into this whole mess of a thing that I can't figure out. But so part of the problem we were having with Arena is we felt like we couldn't sort of see the inner workings of the process well enough. So we couldn't really see exactly where there were bottlenecks. To an extent, you can see the little people running through it. And so these people, that person's waiting to have their EHR reviewed, and that person's waiting to be consented. So, if a whole bunch of people build up, you'll see there's a bottleneck. But you can't see, exactly how long it took patient 1 7 minutes to get their EHR review. You can't see exactly the times for each one, which is something we really wanted to look at. And another thing we wanted to look at that we couldn't quite figure out is since we wanted to follow the doctor, and the doctor was a resource in this, we wanted to try to make it so that you could see like a schedule of first the resource was used to review the EHR, and then it was used to discharge someone, and then it was used to whatever. And these are all features that are possibly in Arena, and someone who's not me maybe knows how to use them, but I didn't get that far with Arena this summer. So to an extent you can see bottlenecks, but not really as much as we wanted to.
When you look at the actual data, how much of variation was there in physician to physician time to complete tasks?
There's actually a lot. So one of the most senior physicians averaged, I think, was about like between 20 and 30 minutes per patient. And some of the younger, because it did tend to be that it seemed like the older physicians who'd been there longer had more of a rhythm down. They could go through patients faster. So one was down to 20 or 30, and others were up above an hour. So that's partially because EGDs can take 5 minutes, and if it's a really complex colonoscopy with a lot of biopsies, that can take, close to an hour. So it's partially that, but it's partially just certain doctors flow through the system at different speeds. One thing we noticed is the doctor who went the fastest and had the 20 or 30 minutes per patient, they would start with a patient and they would look at that patient's EHR and then consent that patient and go all the way through until that patient was discharged, and only after that patient was discharged they would start on patient 2. Whereas other doctors might consent one patient and then do some other medical and then consent patient number 2 and then do procedure on patient number 1, and they would switch back and forth between patients. So that was just a pattern we saw, and it could be that doctor is simply faster and he happens to use that, or it could be it really is faster if you do one patient, then the next, then the next. And that's really the sort of thing we wanted to look at with this, not so much the scheduling, but just how things worked out. We didn't quite get there. Oh, yeah.
And I have a question about how you think these systems, these models should be validated. Because I know you mentioned this 79 versus 53-minute thing, and that's sort of a strategy where you build the model, see how long it takes, and see if it matches real life.
Mm-hmm.
And it seems like another possibility is to try a bunch of different strategies, see which one's better, and then if the model predicts real life, in other words, that you don't care about the absolute numbers, but just comparison of strategies and ordering of them.
Mm-hmm.
Well, what do you think the right approach, the best approach is for these things? In other words, is it realistic to expect?
Yeah, an exact.
You're going to get 79 minutes.
Mm-hmm.
That's the real.
So I've actually now, after doing all this, I've been looking at verification and validation of models, and there's not a lot of literature on it. And what there isn't really one way that's like, this is how you do it, this is right, or this is how close a model needs to be for you to use it. What it sort of says is there's two approaches. One is sort of qualitative and one is quantitative. So qualitatively, they're like, you should just look at your model, and if it makes sense, it's probably good. Or you could get your subject, so for us, the endoscopist, to look at the model, and if it makes sense, you're good. And by that virtue, our model does work. If you watch it, it makes sense how the patients are moving through. It makes sense how the doctor is moving through. But then the other way to look at it is more quantitatively and doing things like t-tests or other statistical tests, in which case our model isn't valid because there's a significant difference between the time it takes for the patients. But I'm not personally convinced that our model doesn't work. I think, maybe if you try how I was saying that our model showed that when you schedule double procedures last, it takes longer for patients to go through. Maybe if they tried that and they found that that is in fact correct. So yes, our model has 20 extra minutes somewhere, but maybe proportionately it still works with the data. So.
Okay, well, I want to thank all of the presenters. Oh, it's already on.
It's actually pretty good. I just had the model. Yeah, it just came out like this week.
Okay, let's go ahead and get started. We are streaming this as we always do, and I will monitor the Twitter feed to see if anyone out there wants to ask any questions, like I did when I tuned into the conference when I was in Riyadh, Saudi Arabia, a few months ago. So today is the first of our summer intern presentations where all of the summer interns get to tell us, and I guess really the whole world, since it's being streamed, what they did during their summer vacation. And this will also be recorded, so they can also point other people to show where they did what they did for their summer vacation. As is usual, I'm going to let the mentors introduce their interns. So I don't know who is going first. The Vishnu. Okay.
So hi, I'm going to be really brief. I'm going to introduce to you Uyen, who is my intern for the year. She's one of our brilliant students and interested in a career in informatics.
So